Hello! Welcome to the Aspirational Neuroscience Journal Club. This time we are covering this truly fantastic paper by Goto et al. Entitled Stepwise Synaptic Plasticity Events Drive the Early Phase of Memory Consolidation. It was published in the journal Science on November 12, 2021. The authors develop a new optogenetic technique which allows them to disrupt with precise timing the early cytoskeletal remodeling phase of synaptic long-term potentiation. They verified that this photoactivation will shrink only those synapses that have enlarged due to learning sometime in the previous 30 minutes. They then use this fantastic tool to dissect the phases involved in inhibitory avoidance learning, arguing that it involves three distinct phases, two involving the hippocampus and one involving the cortex. The Goto et al. Paper builds on a large body of previous research into LTP and uses a number of advanced techniques, many of which were developed by co-authors of the current paper. So I will quickly review these before diving into the Goto et al. Paper's results. First, I will review gross-scale electrophysiological measurement of LTP in hippocampal slices. Then I will review the textbook proposed mechanism of LTP at single synapses. The Goto paper performs a range of their experiments using MNI-glutamate uncaging at single synapses. So I will review two-photon MNI-glutamate uncaging as a method to test the strength, that is, the AMPA current response to a single vesicle release of glutamate at single synapses. And I will review how LTP can be induced at individual synapses via repeated MNI-glutamate uncaging performed in a magnesium ion-free bath. The Goto et al. Paper makes extensive use of the results of this 2014 paper by Bosch et al., which tracked a range of proteins involved in LTP at the single synapse level and on a minute-by-minute basis after LTP induction. We will look at how it tracked the timing of the actin binding protein cofilin and discovered its role in the phases of LTP consolidation at the single synapse level. Goto et al. Fuses the Chromophore-Assisted Light Inactivation protein, it's a CALI protein, SuperNova, to cofilin, and shows that cofilin plus SuperNova allows photo-erasure of LTP in a very precise time window. This sets the stage for the main results. Goto et al. Uses CALI inactivation of cofilin to verify three phases of memory consolidation. Online CALI, performed in the hippocampus immediately after a shock event, erases LTP at CA1 synapses and erases the memory. Offline CALI, performed in the hippocampus during the first day's sleep, also erases the memory. And offline CALI performed in the anterior cingulate cortex during the second day's sleep also erases the memory. This is very cool stuff, tying together cytoskeletal changes in individual synapses to whole animal memory consolidation phases. Let's dive right in. LTP is often studied in hippocampal slices as shown in this diagram. A blunt electrode is used to stimulate a bundle of CA3 Schaefer collateral axons, and an extracellular recording electrode is used to measure the resulting field excitatory postsynaptic potentials in the CA1 area. A single 0.2 millisecond stimulation pulse creates action potentials in the axons, causing them to release glutamate onto the synapses of pyramidal cells in CA1. This results in the solid black curve at the recording electrode. The large spike at the beginning is simply a recording artifact from the stimulus. But this part of the field potential is important. It arises from synaptic currents in the CA1 pyramidal cells. Its slope is a measure of how strong the synapses are, that is, how many AMPA receptors opened to this release of glutamate. During an LTP experiment, a baseline strength for this collection of synapses is first measured. Then a series of high-frequency, 100 Hz, stimulating pulses is given to ensure that both the axons and their target cells in CA1 are simultaneously activated. Then a single pulse is put in again to measure the synaptic strength. The dashed lines here show the CA1 field potential response 10 minutes and 30 minutes after the high-frequency stimulation. The strength of these synapses has increased significantly after the high-frequency stimulation. Here is a typical LTP experiment showing how synapses remain strengthened for 30 minutes and longer following the high-frequency stimulations. Many other experiments have shown that this LTP strengthening occurs at the individual synapse level, and that LTP occurs at excitatory synapses in many brain regions besides the hippocampus, including the cortex, striatum, amygdala, etc. The key point to understand is how LTP in a single synapse is dependent on simultaneous presynaptic and postsynaptic activity, and that is accomplished by the NMDA channel, which I will very quickly review next. Here we zoom in on a single axonal varicosity containing synaptic vesicles filled with glutamate neurotransmitter. And here is a single dendritic spine that is synapsing onto it. The spine contains AMPA channels that open when glutamate binds to them, and NMDA channels that would also open when glutamate binds to them, except that their channel is normally blocked by a magnesium ion. When an action potential occurs in the axon, it releases glutamate. The glutamate binds to both the AMPA and NMDA channels, causing them to open. This allows sodium ions to flow into the dendrite through the AMPA channels, but nothing flows through the NMDA channel because it is blocked by a magnesium ion. But if the dendrite is already depolarized, for example due to a back-propagating dendritic spike caused by its neuron firing, then this positive voltage will dislodge the magnesium ion, which will allow calcium ions to flow into the dendritic spine via the NMDA channel. Normally, the calcium ion concentration is kept very low inside of a cell, so these calcium ions entering have a strong signaling effect, activating calcium-dependent kinases, which go on to phosphorylate proteins, leading to an increase in the overall size of the synapse and an increase in the number of AMPA receptors, thereby explaining the increased physiological response seen in the slice experiments. In a nutshell, this is the molecular basis of Hebbian plasticity, the mechanism that ties LTP to simultaneous pre- and post-synaptic activation. The NMDA channels do the main work by requiring both presynaptic glutamate and postsynaptic membrane depolarization to allow calcium ions in. Papers like these here from Matsuzaki et al., which we covered in a previous journal club, which I will link to below, developed methods to induce and test LTP at the individual synapse level in hippocampal slices. They first patched a neuron and recorded miniature excitatory postsynaptic current events. These miniature EPSCs are the result of one synaptic vesicle filled with glutamate being released at one synaptic site onto this patched neuron. These spontaneous vesicle release events happen frequently and at random in the slice, and as you can see, each generates a current of a few tens of picoamp. Matsuzaki et al. Bathed the hippocampal slice with a compound called MNI-glutamate, which is an inactive form of caged glutamate that can be released at a precise location via a targeted laser pulse. The first thing they did was to verify that they could use this two-photon glutamate uncaging to produce synaptic currents of similar magnitude to those seen in natural vesicle release. Here is an animation that should help visualize this. The two-photon laser is uncaging MNI glutamate in the vicinity of this large dendritic spine, and the resulting AMPA channel openings are recorded at the soma as a small blip of current entering the cell. Matsuzaki et al. Determined the proper laser intensity and duration necessary to give a current response similar to that seen in natural vesicle release. This allowed Matsuzaki et al. To quantify the strength of individual synapses by targeting release of a known quantity of glutamate directly on that particular synapse and measuring the current injected into the neuron. Here is a figure from that Matsuzaki et al. 2001 paper that shows the strength of a synapse is proportional to its volume. Then, in a separate 2004 paper, they removed magnesium ions from the hippocampal slice's back. This meant that the NMDA channels could be opened simply by glutamate binding, since there was no magnesium ions to block the pores. They found that pulsing the laser to deliver the equivalent of 60 glutamate vesicle releases within a time span of only 1 minute could raise the calcium ion concentration inside the dendritic spine to a level that induced structural and functional LTP changes in that spiny synapse. Here they performed this repeated laser uncaging at a single dendritic spine, and almost immediately the spine's volume expanded and remained expanded over the next hour of recording. This increase in volume was matched by an increase in the functional strength of the synapse. I want to stress how cool it is that Matsuzaki et al. 2004 demonstrated the classic slice LTP curves, but on a single synapse level. And in the process, they demonstrated their shared dependence on NMDA channel activation. This technique set the stage for even more detailed studies into the molecular mechanisms underlying LTP, such as this 2014 paper by Bosch et al., which used single-synapse LTP induction to precisely track the time courses of 14 proteins involved in the LTP process. Hippocampal slice cultures were biolistically transfected with a plasmid expressing the target protein, for example the protein cofilin, as shown here, fused with green fluorescent protein. The slice was also transfected with a plasmid expressing a red fluorescent protein in a manner that would fill up the volume of the transfected neuron so that the volumes of individual spines could be measured accurately under the two-photon microscope. Just as in the Matsuzaki et al. 2004 paper, LTP was induced in the target spine over a 1-minute period by repeated laser uncaging. As you can see, the volume of the spine increases immediately, just as in the Matsuzaki paper. But now we can simultaneously track any target protein we like by imaging the green fluorescent signal over time. In this case, the target protein, cofilin, also increases immediately upon LTP induction and remains elevated for the 30-minute duration of the imaging experiment. By dividing the green fluorescent signal's amplitude by the red fluorescent signal's amplitude, the authors could get a measure of the concentration of each protein over time, taking into account the swelling of the spine's volume. The normalized concentrations of all 14 proteins are shown here. The authors used this wealth of data to propose a detailed 3-phase model of the molecular steps involved in LTP. This Bosch et al. Paper is so fantastic and provides so much information regarding the molecular basis of LTP that we really should devote a whole journal club to it. But for now, the takeaway is the important but temporary role that cofilin is playing in allowing the actin cytoskeleton of the spine to grow and stabilize. Guided by this paper's model, Goto et al. Hypothesized that inactivating cofilin during the early phase of LTP would erase LTP by destabilizing the actin cytoskeleton. But inactivating cofilin later would have no effect because other proteins would have, by then, provided additional stabilization to the cytoskeleton. To test this idea, they employed the genetically encoded photosensitizer protein SuperNova, developed in this 2013 paper by Takamoto et al. Reading from that paper, Chromophore-Assisted Light Inactivation, or CALI, is a technique for inactivating target proteins. Stimulation with intense light irradiation yields short-lived reactive oxygen species, ROS, such as singlet oxygen. ROS damages proteins close to the chromophore. Since the half radius of photodamage of singlet oxygen, 3 to 4 nanometers, is smaller than the mean protein-protein interaction distance inside a cell, specific proteins can be inactivated by this method, whereas neighboring molecules remain intact. Goto et al. First biolistically transfected hippocampal slice cultures with plasmids expressing the cofilin SuperNova protein, as well as a red fluorescent protein to fill the volume of the cells. The authors included a cofilin GFP fusion protein as well, so that they could track how the disruption of cofilin SuperNova affected the concentration of other cofilin molecules. At 0 minutes, LTP was induced, as in the previous paper, using repeated two-photon MNI-glutamate uncaging in a magnesium ion-free bath. Like previous papers, the spine increased in size almost immediately. But after 10 minutes, the authors used a different laser wavelength to cause immediate destruction of the cofilin SuperNova proteins. As the authors had hypothesized, this reversed the LTP enlargement, and that particular spine shrunk back down to close to its baseline size. The authors verified this CALI-induced shrinking did not permanently damage the spine's ability to undergo LTP by successfully inducing LTP in the same spine 20 minutes later. This can be seen in the orange curve in this plot. Initially, the spine's volume increases after the LTP induction at time 0. Without CALI, the spine's volume would decrease somewhat, but would remain elevated even 60 minutes later. If CALI is induced at time 10 minutes, the spine's volume quickly returns to its initial size, thus erasing the LTP. To test whether this permanently destroyed the spine's ability to undergo LTP, the authors then induced LTP again in the same spine via MNI-glutamate uncaging at the 30-minute mark, and the spine's volume again enlarged and remained enlarged just as in normal LTP. So the CALI does not permanently damage the synapse. The authors then sought to determine the time window that this CALI-induced shrinkage was effective over. CALI 1 minute before LTP induction did not erase LTP enlargement, and neither did CALI 50 minutes after induction. But CALI performed between 10 and 30 minutes after LTP induction did erase LTP enlargement. Recall that this limited window of cofilin vulnerability was precisely what was predicted by the Bosch et al. Model. Encouraged by these successful single synapse results, the authors then created an AAV viral vector containing the cofilin SuperNova gene and used this to infect the CA1 region of a hippocampal slice. This way they could test if LTP could be erased in large numbers of synapses in the classic slice electrophysiology experiment. Then the authors performed the standard CA3 Schaefer collateral to CA1 hippocampal slice LTP experiment that we discussed previously, and tested whether performing CALI-induced cofilin supernova destruction at 10 minutes after high-frequency LTP induction would erase the potentiated electrophysiological response. It did just as expected, as seen by the red curve here. With this result, the authors had shown that this cofilin supernova viral construct could provide a way to erase LTP at particular synapses in a very precise time window, the perfect tool with which to study the memory process in living animals. The authors then injected this cofilin supernova viral construct bilaterally into the hippocampal CA1 regions of living mice, and implanted optical fibers above both hippocampal regions so that CALI could be initiated at precise times. The mouse was then trained in an inhibitory avoidance task. The mouse is first placed in the lit side of a partitioned chamber. There is a door to the dark side of the chamber, but it is initially closed. At time 0, the door is opened. Because mice prefer to be in the dark, they will typically cross over to the dark side within about 30 seconds of the door opening. When the mouse crosses into the dark side, it gets shocked. 2 minutes after this shock, the experimenters induce CALI to attempt to erase the recently potentiated synapses in the hippocampus that supposedly encode this fear learning event. The authors refer to this delivery of CALI immediately after the shock event as online CALI, to distinguish it from later experiments where CALI is delayed hours or days. This sequence of events is displayed diagrammatically here. On day 2, they put the mouse back into the light side of the chamber and then open the door at time 0. A typical mouse will hesitate much longer before going into the dark side of the chamber because they remember the shock from the previous day. However, if the experimenters were successful in erasing the memory with CALI, then the mouse should enter the dark side of the chamber just as quickly as they did on the first day. In some experimental cases, the mouse is shocked again on this second day's entry into the dark side, so that its memory can again be tested on the third day. And here are the results. A control mouse that had no surgery crossed over into the dark side just seconds after the door was opened on the first day and got shocked. And when presented with that same option a day later, the mice hesitated on average 400 seconds before crossing over, showing that they had a strong memory of the shock. In contrast, cofilin plus supernova mice crossed over into the dark side on the second day about as fast as they did on the first day, showing that the memory of being shocked had been erased. This forgetting was specific to that first CALI erased memory. It did not negate the ability of the mouse to learn this avoidance task. We can see this because on the third day, the CALI mice did hesitate before entering the dark side, showing that they had formed a new memory from the shock on the second day that was not erased. The authors performed this same experiment but with a range of time delays between the shock and the CALI erasure step. CALI was effective at erasing the memory only if it was delivered within approximately the first half hour of the shock. Recall that this is the same time window of effectiveness that was seen in the single synapse experiments. Next, the authors asked if this erasure of memory was specific to the context the shock was experienced in. Other studies have shown that hippocampal-based memories are especially sensitive to the place where the memory was formed. The authors put a mouse first into the context A chamber, depicted here as a chamber lit by green light and having a star figure on the wall. When the door opened to the dark side of the chamber, the mouse quickly entered and was promptly shocked, developing a memory for this context A. 2 hours later, the same mouse was placed in the light side of a different chamber, context B. When the door to the dark side opened, the mouse again quickly entered and was shocked again. It was not afraid in the context B chamber because it had never been shocked in context B. The experimenters then used CALI to erase this memory of the context B shock. Here are the crossover latency results for both control and CALI mice, showing quick entry for both contexts in these initial exposures. On the next day, the experimenters put the mouse back into context B, and when the door opened, tested how long the mouse hesitated before entering the dark side of the chamber. No CALI control mice showed a significant delay, indicating that they remembered the shock the day before. But the CALI mice showed significantly less hesitation, indicating that the CALI was at least partially effective in erasing this memory. 30 minutes later, the mouse was put back into context A, and both the CALI and control groups showed long hesitation times before entering the dark side. This proves that the CALI-induced erasure was specific to the memory formed in context B. It did not erase the previous memory formed in context A. A large number of studies have shown that hippocampal replay during sleep is important for memory formation. Given this background, the authors developed a method to precisely monitor a mouse's EEG and EMG signals to automatically determine if the mouse was asleep. They then applied CALI to the hippocampus either only during awake periods or only during sleep periods. The black lines in the plots show the sleep state of the mouse, and the red lines shows times during the first 7 hours when CALI was applied to the hippocampus. Mice who had CALI performed during their first day's sleep periods showed significant erasure of memory. But if the same quantity of CALI was performed while they were awake, there was no significant memory erasure. And CALI during sleep on the second day was no longer effective in erasing the memory. This is a truly amazing result. CALI performed in the hippocampus during the first day's sleep period erases memory. The authors perform additional experiments, which include calcium imaging of CA1 neurons during the task, in an attempt to explain what is really occurring during the first day's sleep in the hippocampus. To be honest, I found these experiments difficult to understand, and the results difficult to interpret, so I won't cover them in detail here, except to point out that they found increased synchronization of firing after the first day's LTP. This suggests that LTP during sleep has made the hippocampal representation more robust and potentially more effective at driving other brain areas. Personally, I think the most straightforward way to think about this offline CALI erasure during the first day's sleep is to understand that the hippocampus replays the memory in a manner that allows further LTP of those same synapses in order to better consolidate the memory in the hippocampus. However, in order to strengthen these synapses, they must be made liable again. So CALI during these sleep replay events can erase the original memory. In my opinion, this interpretation is more in line with papers like these shown here, that show that recalling a memory makes it vulnerable to erasure. This effect is called amnesia caused by disruption of reconsolidation, and there is a substantial literature on it. Finally, the authors explored how a memory that is initially formed in the hippocampus is consolidated in the cortex. Clinical evidence and animal studies have long shown that the hippocampus is only the initial encoder of a contextual fear memory. If a memory is to last weeks or more, it must be re-encoded into the cortex. Articles like these, shown here, suggest that regions of the prefrontal cortex, like the anterior cingulate cortex, are crucial to this, and suggest that the process of transfer from hippocampus to cortex occurs via hippocampal replay during sleep. With this background, the authors next injected the anterior cingulate cortex of mice with the cofilin SuperNova viral construct and implanted optical fibers to illuminate the anterior cingulate cortex, as shown here. CALI was performed in the anterior cingulate 2 minutes after the mouse was shocked and was completely ineffective at erasing the memory, which is expected since, again, the hippocampus is known to be the initial encoder of these contextual memories. Then CALI was performed in the anterior cingulate cortex starting 2 hours after the shock for the following 8 hours. This was also completely ineffective at erasing the memory. Recall that this would have been effective to erase the memory if it had been performed in the hippocampus, since it would have coincided with many first-day sleep periods. Finally, the authors performed CALI in the anterior cingulate cortex on the second day after the shock, and they found that this time the memory was erased when tested on the 3rd day. The authors also compared CALI during awake periods versus during sleep periods and found that CALI was only effective at erasing the memory when applied during the sleep periods of the 2nd day. This may seem like a bizarre result to the uninitiated, but it actually fits quite well with the existing literature on remote memory consolidation. The cortex is the ultimate holder of such memory, but its synapses must be trained by hippocampal replay events during sleep. This means that there must be some times during which cortical synapses are undergoing LTP. The authors of this paper simply identified those times, and it turns out that CALI during the 2nd day's sleep was highly effective at erasing the memory. Presumably, erasing LTP in anterior cingulate cortex earlier would not work, since the hippocampal memory was still strong then. Let's hear how the authors summarized their results. Quote, "We developed a versatile optogenetic method allowing for the selective optical erasure of LTP in a spatially and temporally restricted manner. We found that hippocampal LTP occurs in 2 distinct temporal processes: online, immediately after learning, and offline during periods of sleep. After 2 waves of LTP in the hippocampus, a 3rd wave of extrahippocampal LTP takes place during sleep the next day in the anterior cingulate cortex, and this is required for systems consolidation." I hope you agree that this is some truly fantastic neuroscience research. To me, what is super important here is how clearly this work shows that whole animal, behaviorally relevant memories are implemented in structural changes to specific synaptic connections. If you reverse the structural changes to those specific synapses by destabilizing their cytoskeletons, then you erase that particular memory in the whole animal. Thank you for listening.